all kind of analysis. For me, it means more like an analytic, which is a combination which definitely has a lot of number crunching at the end and a set of code for that before that. But even before that, you need to have a visualization of what you're going to do, what is going to be the strategies, and those are, I think, the major core steps in predicting, I think, as a part of the analytics process. So here I showed some couple of the slides of the work that I do as a bioinformatician at the internet. So if you look at an individual genome, which is of a sick person or a healthy individual that is sequenced, and then compare it to a reference genome sequence, such as the build 37 sequence we have, we can get a lot of variation. That can be an insertion, that can be deletion, an inversion, or copy number variation, and single nucleotide polymorphism. And all of them can be related back to another database which is a record of which variation has been found already in diseases to a different patient. So we can always correlate with that. And of course, it's a two-way process. That database builds it up, and this also keeps on recording it. So one of the thing is that we need to have good assembly to be generated, and this is just the bioinformatics result that people were using slope de novo software. That is a yellow bar, which is it's quite fast. But there are other tools which are slower, but it gives much better results. That's the pink bar over there. But the scalability is better for the slope de novo software. So this is just one of the bioinformatics software results. So here's an example. Sorry, it's not so clear, but we have A105A as a father, B as a mother, and they have two children. And I assembled on the y-axis is the genome of assembled genome of each one of them, and on the x-axis is the reference genome. And I made a software using a combination of this analysis or genome break that essentially finds out the break in the genome of the candidate sick person or maybe the individual as well, to know where are the variations. And once we know that variation, as I've been telling again and again, we can trace it back to any database that will maintain record of which loci in the genome, which genes are related statistically. These are all predictions to any disease. Or we can also do scientific findings, as I'm doing it over here. So, for example, I took the mitochondrial genome, which was known to be inherited totally by maternity, which would mean that any structural variation, any SNP that is present in the mother should also be present in both children. And anything that is found in the father should not be present in probably the children if it's found in the mother. Whereas I've got evidence over here where one of the structural variations has been found in the father, and it's found in the children as well as found in the mother. And on the lower axis is the total sum of the total structural variation basis and the sum of the SNP basis, which also tells that even in one generation you can have quite a lot of mutations happening. So this is the next slide. The previous one was for structural variation, and this is for SNP. So the green colored ones are the ones. So the left one is A10A, which is the father, and A1053C is the children. They both share a common SNP in the mitochondrial DNA, but the mother doesn't have it. So it can be a clue that our knowledge that maternal mitochondrial DNA is maternally inherited totally might not be the case. We need to do further investigation and maybe further more validation experiments to confirm whether this is the case. So this is the power of prediction that we can perform. Another great application I'm telling you that people using different technology, ultrasound or MRI scans to find out the fetal sex of the baby. Here's another technology. Anyways, we are going to move in a personalized genomic medicine age where everyone is going to carry their genome in a chip because it's going to be just a few $100s to get your genome sequence. So why not do it when you're in the fetal sex and at that stage structure variation basis of each of the individuals, what we see that the father on the Y chromosome has far higher number of structural variations than the other three persons, the mother and the two children, confirming that the two children are daughters. Because the woman doesn't have a Y chromosome, but there are Y chromosomes crossing over, so they might have parts of Y chromosome and other chromosomes. And another finding over here is that the structural variation is far more restricted. There is more selection pressure on the structural variation than on the single nucleotide polymorphism. So structural variation is more a fingerprint, a more stronger characteristic of an individual than a sense. So, and can be linked directly to the individual. So these are two new discoveries, or I would say one technology and another discovery. So to sum it up, the conclusion would be that genome-wide fingerprints can be determined now at a much cost-effective rate, because the cost of sequencing has been going down and down rapidly and improving in terms of technology merit, because now we have paired sequencing. Earlier we had just one sequence end. Now we have paired it with the rough synthesized node, so that's going to be extra information which we can use. And we can now do sex determination as well. There's a possibility, as I said, for doing new discoveries, mitochondrial DNA leakage. Another point that I did not show over here, but I'm telling you, that we know that the HLA domain, which is the major histocompatibility domain, is found in chromosome 6 of human beings. If we can have a record of that in every individual, that these are the structural variations, these are the single nucleotide polymorphism of the chromosome 6 of that individual, then here are the possibility of organ transplant or any neurological response for that individual. And the final discovery that I made is that the structural variations are more specific and characteristic to an individual than SNPs are, because they have more selection pressure. And of course, these variations can be linked with another database and other relational database that we have been talking throughout our conferences, integrating different databases to make statistical measures that these sequences might be related to these kind of gene mutations and other kinds of symptoms. With this, I would like to conclude, and that's my email address. Please do get back to me and be in touch.
Thank you.